All right. So my presentation or my project is about the role of AUF1 in messenger RNA decay in idiopathic pulmonary fibrosis. So for starters, oops. Okay. So for starters, idiopathic pulmonary fibrosis is essentially a disease which involves the progressive and accelerated aging and scarring of the lungs, which can damage the lungs' capability for gas exchange and eventually leads to respiratory failure. So the scarring in IPF can be attributed to the excessive collagen production or excessive protein production in the lung's extracellular matrix. So one of the reasons why this happens is because of the stabilized decay rates of messenger RNA transcripts. Now, stabilized decay, what that means is that the transcripts are decaying at a slower rate. And the reason why this is really important is because transcripts, messenger RNA transcripts, are responsible for transcribing and synthesizing proteins. So when they are decaying at a slower rate, they're around for an abnormally long period of time, and they also produce more proteins than they should be producing. And so for a visual, you can kind of see on the left we have an IPF lung, and on the right is a normal lung. So on the left, the first thing that kind of comes to, at least catches my eye, is sort of these honeycomb-like areas on the IPF lung versus the normal lung. There's no such area. And so the reason why this is because of fibrosis. So this is because fibrosis, or the excess collagen production, is happening in the ECM, which is what's causing these these shade differences. Another thing that you have to look a little bit harder to understand is this sort of black area. As you can see, the rest of the lung is gray, and I don't know if you can see my mouse, but here the rest of the lung is gray, and then the normal lung is also pretty gray. But here, if you look, this area is more black, and this is because of scar tissue. So this area is essentially devoid of lung tissue, and they're air sacs, which is why it appears as black on the CT scan. So I was mentioning earlier the extracellular matrix, and that is where the fibrosis happens. So essentially, we're focusing on the protein fibers, and this excess collagen is what's building up and is being produced, and that's being produced by the fibroblasts. So one sort of element that's already been identified by previous research is the dicer1 transcript. So dicer1 is an mRNA transcript that is involved in the regulation of fibrosis in IPF. So it's been identified in a feed-forward loop in which lung fibroblasts can actually sense the fibrotic ECM, and this causes dicer1 to have destabilized decay, which means that it decays at a faster rate. And because dicer1 is involved in regulating fibrosis, when there's less dicer1 around, that actually causes an increase in ECM deposition and progression of fibrosis. And that further leads to less dicer1 being present and less dicer1 regulating fibrosis. And so it's sort of this like feed forward loop that occurs in which there's more fibrosis and less dicer1 present. So for kind of a visual, you can see here, this is the microRNA 29 complex. So dicer1 kind of binds with this and works with microRNA 29. And so when the fibrotic ECM, fibroblasts can sense the fibrotic ECM, and that further leads to more fibrosis and less microRNA 29 complex being present. So another element that's already been identified as something that plays a role in IPF is RNA binding protein AUF1. So it's also known as RNA binding protein AU-rich element binding factor 1, which is sort of a mouthful. And the hypothesis here is that essentially the post-transcriptional binding of AUF1 actually leads to destabilized messenger RNA decay, which means that these transcripts are decaying at a faster rate. So they're not around as long. And this can lead to the development of the fibrogenic phenotype. So the reason why AUF1 is even connected to IPF, and this is sort of where the scholarly work that's already been done before comes in, is the expression of the dicer1 transcript is actually controlled post-transcriptionally by AUF1. So when AUF1 binds with dicer1, it actually leads to the rapid decay of the dicer1 messenger RNA transcript. And This further leads to the expression of the fibrogenic phenotype because dicer1 is involved in regulating fibrosis. So the question that I'm addressing is really, what is the link between AUF1 and IPF? So which messenger RNA transcripts exactly have a stabilized decay rate in the absence of AUF1 on the IPF lung matrix? And are there commonalities between these transcripts? So the hypothesis that we have is that the post-transcriptional binding of AUF1 to messenger RNA leads to more rapid messenger RNA decay on the IPF lung tissue and thus to the development of the fibrogenic phenotype. So for the process of research question selection and how I even came to this topic, because it's quite a specific topic, I first started with just doing background research on dysregulated cells and understanding cell death disorders. And that was really what my main interest was. This, led me to go a little bit more into the cancers, and understanding more about like sarcomas was another one of my interests. And then I also found out about IPF through this background research and reading scholarly work. And IPF immediately sort of drew me in because it was something that I hadn't heard before and I hadn't learned about much before, versus cancers and the sarcomas that I was interested in, I had learned about before. And so that's sort of why I chose to do more work on IPF and learn more about it. And a lot of the type of research I did was also dependent on the pandemic and sort of the work opportunities that were also available during the pandemic and could be done remotely. And so that's sort of where my mentor's lab also played in. And I was able to do a lot of analysis work with messenger RNA decay. And that is sort of where that's how I selected my question. So for my method, I used a mixed method, so it had both quantitative and qualitative aspects to it. So for starters, human lung tissue was procured and de-identified by the University of Minnesota. And the data was sourced through 4 different conditions. So what's really important to understand about this lung tissue is that it was decellularized, which means that the extracellular matrix was actually isolated from its inhabiting cells. So it was just the ECM that was part of this tissue. And then fibroblasts were then cultured on this ECM. So here's sort of a diagram which explains what exactly we did, and it can explain the 4 different conditions really well. So we have, the IPF ECM or the diseased ECM, and then we had a control ECM or a healthy ECM. And then we had fibroblasts that had AUF1 present that we added to both the IPF and the control ECM. And then there was fibroblasts that had AUF1 knocked down or AUF1 not present. And then those were also added to the IPF ECM and the control ECM. And so we have 4 different conditions. So after this, the messenger RNA transcripts were all measured at different times, or their abundances were measured at different times. So for example, they were measured at time 0, time 90 minutes, time 180 minutes, 270 minutes, etc. And all of these abundances were then added to a table. And this is really how we calculated the decay rates. And so this is where my work with the data analysis began. And I used the R program, and I converted this data into a linear regression line via a logarithmic test, and I used a log-base 2 function. Afterwards, I did a statistical test which could tell me which messenger RNA transcripts specifically had stabilized decay rates in the absence of AUF1. And I did this for both the control and IPF matrix. And this was, up till now, it was sort of more of the quantitative method, because once I did the statistical test, R gave back a specific number of transcripts which were involved or AUF1 dependent. And so then more of the qualitative work began, and in which messenger RNA transcripts were analyzed for enriched gene ontology using the Gorilla server. And what this essentially did was it found common processes shared between the messenger RNA transcripts in each list. And it also gave like respective p-values and FDR values for these common processes. So of these common processes, first, these were the results that R gave back after the statistical tests. So these were the messenger RNA transcripts that were dependent, that were AUF1 dependent. So on the control matrix, we found that there was 1,178 mRNA transcripts. On the IPF matrix, we found there was 1,642. And between the two, there was only a commonality of 97 messenger RNA transcripts. So after we did the gene ontology, so we took these lists of transcripts and we put them into the Gorilla server. And I was able to find that and this is kind of the results for the IPF transcripts, or the transcripts that were dependent on AUF1 on the IPF lung matrix. So one of the proteins, or the protein coding transcripts that shared a lot of similarities with the transcripts on the IPF lung matrix was HNF4A. And this is essentially a transcript which controls a lot of hepatic genes, so liver genes. And a lot of the mutations in this transcript were also associated with monogenic, autosomal dominant, non-insulin dependent diabetes type 1. Diabetes type 1 is an autoimmune disease. And so this gives us a lot of insight into the type of, the role that AUF1 plays in IPF and how it may have some autoimmune aspects that are sort of adding on to IPF. And also on the IPF ECM, for the transcripts on the IPF ECM, there were some other processes included cell adhesion, including adhesion proteins and cell membranes, as well as organismal injuries and abnormalities, as well as cell death and survival and cell death disorders such as cancer. So adhesion is important because a lot of adhesion proteins and cell membranes are involved in the ECM as well. And so, fibrosis happens in the ECM. So again, this shows that AUF1 may play a role in fibrosis through mRNA decay. Some other organismal injuries and abnormalities, cell death. Cell death is also a huge one, and it's not unexpected that these transcripts share a lot of similarities with cancer or other cell death disorders, because we do know that IPF and cancer are quite similar in the way that they are both progressive diseases that often go unchecked. So on the control matrix, there was two main processes that we found. The first was the cellular process. And now this is quite a broad term, but it refers to cell communication, cell movement, cell proliferation, as well as cell specialization. And this is sort of what the AUF1 dependent transcripts do on a healthy lung. Another process was the regulation of cell death. And as you may recall, in IPF, the IPF transcripts that were AUF1 dependent were also involved in the regulation of cell death. So on the healthy lung, these transcripts are essentially they're essential for tissue homeostasis, and we know that on the IPF lung, tissue homeostasis is not happening because of fibrosis and because there's excess collagen and protein production happening. So again, that sort of shows us the role of AUF1 in the regulation of cell death, and opens up, more possibilities for research in the future regarding AUF1 and its role in cell death. So for the 97 overlap genes between the control transcripts and the IPF transcripts, there was no common processes identified by the specific server for gene ontology that we were using. So in conclusion, we did accept the hypothesis that the presence of AUF1 did destabilize certain transcripts. And another really important key takeaway from this project was the fact that the diseased ECM actually has the capability to change a cell's phenotype. And the reason we know this is I mentioned that this data was, or this tissue was decellularized. So it was just the ECM that was taken. It was either diseased or IPF ECM or healthy ECM or control ECM. So between the two, we know that because it was decellularized, it meant that it had no cells, and the fibroblasts that were cultured had only the environmental signals to go off of. And because we know that in the IPF ECM, it still acted as an IPF fibroblast, and fibrosis still happened. And so we know that it took on environmental signals because of that. And so that tells us a lot about fibroblasts and the fact that the disease ECM can change a cell's phenotype. However, there were also shortcomings within this project. The first one, and the main one, being that we inferred that these transcripts were AUF1 dependent transcripts because we couldn't actually observe these cells. So, or these, yeah, these cells and these transcripts, because that would require a lot of resources and a lot of cells and technology that we do not have yet. Another shortcoming was that the gene ontology servers didn't recognize all of the transcripts. So for example, out of the 97 common transcripts between the transcripts on the IPF and the transcripts on the control lung, only 67 of them were identified. Now this may affect some results, but the probability that it does affect the results largely is really low because it was out of the 97, because 67 were identified, that's the majority of them, and only 30 were not identified. And 30 transcripts in this case would not make that much of a difference, especially when you're considering p-values and FDR values, or false discovery rates. So the implications of this project, so it really connects AUF1 to cell death and cell adhesion, mostly in IPF, as well as on the control matrix. We know that AUF1 is connected to cell death. And that opens up a lot of research opportunities in the future to understand how exactly AUF1 may be related to cell death and cell adhesion and all of those processes that I mentioned before. It also connects IPF to other diseases. So as I mentioned, HNF4A, which is responsible for type 1 diabetes if it's mutated. And because we know that type 1 diabetes is autoimmune, we also can make inferences that IPF may have some autoimmune aspects to it. And also it connects IPF to cancer and other cell death disorders because we know that these diseases are going unchecked. And it kind of reinforces sort of information we already had about IPF going unchecked. So thank you. That was sort of it for my project. So thank you for listening, and thank you for your time.
I was distracted momentarily from a fire drill, and I'm not sure, was that the end of your presentation and now you're ready for oral defense questions?
Yes, yes.
Okay. I am not sure how much time we have remaining because my timer went off, but I'm going to ask these questions and you can answer them. Ready, Maria?
Yes.
All right, here we go. My first question for you. How did the choices you made when designing or implementing your research method impact your research process?
So one of the main choices that I had to make was because of the pandemic, and I think the pandemic has impacted us in so many ways, but because of that, I had to do a type of research that was remote. And I think because of that, I chose to do data analysis. And a lot of that had to do with also my mentor's lab and the work that he was previously doing. And so, and that's why I really chose to work with R and study messenger RNA decay specifically. And obviously that impacted my whole research process because that was kind of this the majority of my methodology. And it also, I think I learned a lot from it, and a lot from the data analysis part, because I learned skills that I had never had before. So yeah.
You may have already addressed this, but my next question is, how do your findings provide direction for future research in this field?
Yeah, so well, my findings really, what they did was they connected AUF1 with messenger RNA decay in IPF, but I think there's still a lot of research yet to be done on how exactly AUF1 is connected to the specific processes that these transcripts were involved in, and understanding that further in detail. And that's, I think, something that can be done in the future.
And then finally, I am curious to find out more about how you handled uncertainty in the research process.
So and again, I was so lucky to have Dr. Bising as a mentor. I think the main way I handled uncertainty was either, doing more research or asking questions. And I think asking questions was really like the big, especially like online. I think it was definitely harder for me to like understand certain concepts, and working with something that I was completely unfamiliar with was with R and data analysis. I definitely had a lot of uncertainty, but asking questions and specific questions was really what helped me. And also making mistakes was something that helped me learn and understand that making mistakes is okay, and that's totally part of the research project or process. So yeah, that is how I handled uncertainty.
Outstanding. Well, that is the end of the formal part of your presentation with oral defense. Stop recording.